So I hope you'll forgive me for the kind of rather grandiose title of this talk. As it turns out, the talk really has two parts, and the first part really is very, very specific. It's work that we did for a period like about 7 years. It was just absolutely delightful collaboration on building building synthetic devices or synthetic shapes based, in a kind of bio-inspired way, with incredibly creative experiments being done in my friend David Gracias's lab at Johns Hopkins. And in some sense, what I'm really trying to do is to, in part resurrect this work, but in part the work has kind of sat there for a little while. So, there's a clean mathematical framework and a sense of kind of solid mathematical understanding. And I want to understand if I can actually scale that. And so kind of like the last part is really, in some sense, a question which is in some ways actually intrinsic to mathematics. And I kind of want to explain how I kind of started with something which is very biological and end up with something which is, in some sense, for me, foundational mathematically. So, Bernard in his first talk kind of had mentioned that, if you really want to understand problems, a great deal of physical understanding comes from model problems where we have a lot of symmetry. And so I'm going to first explain to you what self-assembly with a lot of symmetry looks like. And first of all, to introduce the term self-assembly. So one of the first uses of the phrase self-assembly is by Casper and Klug in their work on the structure of viruses. Very, very early stages of molecular biology. And it's the work that followed the second paper that Watson and Crick wrote after the first paper on DNA was about the structure of viruses. They found that viruses have icosahedral symmetry. And this idea was really kind of, just really studied with great care by Casper and Klug. And so they put forward something which is dogma, which we will criticize later. But I just want to put it up front. This was a movement of physicists towards biology. And so at the time, what they thought was that statistical mechanics was the right framework for thinking about this. So the reason they came to the problem in this direction is that this type of virus is the kind of structure that they had seen. And when you see structures of this nature, they're very symmetrical, they're beautiful, and you think that you should be able to understand that. And that's something that we would like to do. This structure, C60, was discovered only in the late 1980s in the work of Croteau and Smalley. And here's a kind of other example of a zooplankton, which has icosahedral symmetry. So what mathematics allows you to do in these problems is to focus on the shape rather than focusing on the scale and the underlying physical mechanism. So that's going to be a theme of this talk. I want to kind of focus on geometric principles, to kind of use minimal geometric principles to try and understand how these things build themselves. So the work that I was doing is actually on synthetic self-assembly. So what you really want to do is you want biology to inspire the design of devices and materials on very, very small scales. Biology has just solved incredibly hard physics problems on scales that are absolutely fascinating. And so, in the study of models, of course, you hope that the study of these models will somehow shed light back again on biological systems. So when you're building stuff, you kind of want to abstract. So you want to have some stripped down interactions. We're going to begin with simple shapes out of a few simple motifs, and then add in a little bit of randomness. Okay. So around this period, like 2010, 2000, there was a kind of explosion. A bunch of different people began to build polyhedra in all sorts of interesting ways, based upon kind of whichever discipline they were in. So these are two examples from supramolecular chemistry. So what you're actually doing is you're taking agglomerations of particular molecules. So in this case, it's kind of organometallic supermolecules. So you kind of build up a complex. It's got, these are not real elements. It looks like a chemical formula, but it's not real elements. What you've done is synthesized a kind of metal piece and a ligand piece, and you put them together in a rather creative way to build cages. Okay, so the shapes over here are primitive shapes, and so it's very natural to kind of try to build these structures. So here's another one. So this is it's part of a field that I'm going to kind of refer toward the end, the field of kind of DNA origami. But this is work from Yamuna Krishnan's lab. She's now at Chicago, but this is back in the day when she was at the National Center for Biological Sciences. And what she did was to kind of create kind of synthetic cages using DNA. So what's really going on over here is you take a shape, you break it up into fundamental units. So over here we're thinking about the icosahedron. So it's made up of triangles. I focus on each edge of the triangle. Each edge of the triangle is actually made with DNA. And these orange bits over here, they reflect kind of complementarity. So you have a DNA with a little bit of a sticky end. You throw it into a heat bath. These sticky ends mate. And you kind of make a chemical reaction out of DNA. OK, so polyhedra have been around in math for a really long time. And there's a kind of delightful book by Hugo Steinhaus from the 1930s, where he's talking about shapes. And this is a lovely book for lay people. And what Steinhaus does is to kind of build a polyhedron in a slightly different way. What you do is you unfold the polyhedron. So imagine cutting it in a particular way. So that's the final three-dimensional shape. This is a two-dimensional shape. The two-dimensional shape is called a net. It's made up, as you can see, of kind of contiguous elements. And what you do is you kind of take these contiguous elements, you put it together, you have a rubber band, and up it pops. So you kind of use physics to get this sheet to make itself. OK, so this is stuff that we designed at Brown and was built in David's lab. And I'm going to let you just watch the movie. This is on a micron scale. So that's about 3 hairs width. And you're looking at it under a microscope, and it's going to make itself. OK, so that's an example of synthetic self-assembly. I feel a bit like an aging rocker kind of trotting out greatest hits. But this is really one of the best things I've done in my life. And I want to say yeah, James.
What's it speed up like on that video?
Sorry?
What's it speed up like on that video?
That's real time, no speed up. That's exactly the real scale. So I don't want to kind of get into the physics of this in the sense like what's actually happening over here is it's driven by surface tension. The long story is that photolithography allows you to stuff that was used for the computer industry. So people have very sophisticated ways of kind of manipulating thin films. David, over a period of 10 to 15 years, developed this technique for kind of putting on solder on these edges, and then it's in a hot solvent, the solder melts, the thing moves because of surface tension. Okay? So in a sense, what I want to do is we developed a theory for understanding stuff of this nature, but when you're kind of developing a theory of stuff of this nature, this was inspired by biological context. So I want to give you a sense of both the biological context as well as the kind of mathematical framework. So that's the question, right? So can we develop common frameworks to understand biological and synthetic self-assembly? And our approach is going to be very minimalistic, because the reason you want to be minimalistic is because you want to get a theory that is kind of scale invariant. So you kind of want kind of simple common principles, easily implementable. And so what we're really going to do is I'm going to introduce these terms, model intermediates and pathways of assembly. So that's what I really want to understand, the process. How does this thing actually happen? And I want to understand the most robust features of simple models. So the geometry is the most primitive part, and it turns out that models of this sort had been introduced by a virologist, Zlotnick, and it was reintroduced by a group of chemists, and they called it the building game. And so the game is, how do you build a shape? Okay, so you can think about it, for a mathematician, this is like a coloring game. So you start out, so this is a representation of the dodecahedron. It's something called a Schlegel diagram. It's a way of seeing the three-dimensional shape in 2 dimensions. And you can see exactly what's going on. At each step, I form a bond. And the rule is that, if you've got a particular cluster, you choose where to put the next one, right? So you kind of discretize the entire pathway, this continuous way in which stuff is happening with a collection of shapes. So we've discretized the assembly process into a bunch of intermediate states. So that's the kind of final product, but the way you get to the final product is by going through these states. And so if you're doing something of this nature, you need to also have the physics to kind of say how you have attachment and detachment. But we are separating the geometry from the physics. So the geometry is, for us, the most primitive object, and that's what we're going to focus on. So this idea is very useful in chemistry for modeling fullerenes. It has been used for viruses. That's where Zlotnick began. But it doesn't explain, for example, malformed shapes. It's like, it tells you, like, here's a pathway to the complete object. But you may, of course, screw up in zillions of ways. And it has no sequence-specific information. So that's something that I'll kind of come to in a little bit later. So this idea actually, one of my favorite papers in this area is a sort of loopy paper by this guy Hosokawa and people around him. And so what Hosokawa did was to say, okay, what's a macroscopic chemical reaction? So what he did was to make these little, oh, great. I seem to have lost, oh yeah, there we go. So he made these little triangles, put little magnets into these triangles, put it into a box and just like shook the box. And what happens is, of course, you formed some complete hexagons that you're looking for, and you've got a whole bunch of other stuff too. Okay, so this experiment is very interesting because somehow you're doing chemistry without doing real chemistry. And you're doing chemistry in the sense that you have chemical reaction theory. Okay, so chemical reaction theory says that, okay, you break up the process of assembly into partially formed pieces. You have chemical reactions that tell you how you go from one state to the other. So take 2 units of X, and you'll get X2. Take a unit of X, take a unit of X3, you'll get X4. So you make a bunch of reactions. So the reactions are based on the underlying kind of combinatorial decomposition. But you need to add rates to these reactions, and that's where you really do have to do physics. So somehow there's this part which is actually quite robust. This part is actually less robust because this is where you really have to make modeling assumptions. What sort of magnet were you using? Are you sure you can ignore friction? Are you sure you've got the geometry right with collisions? So this part, in some sense, is as subtle a problem, but that part is really, really robust. And so you can kind of use the robust part, understand that really well, and use it as a scaffolding to add in new ideas to your theory. Okay, so what that actually means, so as mathematicians, we like structure. So what that really means in terms of structure is that we've taken, so this is actually for the building game. So there are different discrete models, and I use them in different ways. So this is for the building game. So I'm building the tetrahedron. These are all the possible pathways and all the possible intermediates that you can make for the tetrahedron. So there's a mathematical problem over here. The mathematical problem is take a particular shape, break it up into pieces, ask yourself what's the number of pathways that you can get to go from point A to point B. So that's one piece. It's an assembly graph. And then there's a second piece, which is kind of quantitative information that you layer on to the assembly graph, and that's really equipping each edge over here with a rate. Like, what's the likelihood that you're actually going to take this path or that path? Okay, so that's the model. Okay, so let's take this model and let's go back to where we began with viruses, and let's take a look again at how this model has been used. So this is not quite my work. This is work of Raidun Twarok and people around her, but it's really, really interesting work, and I think to share it with a broader audience is good for everyone. Okay, so viruses are super interesting from the viewpoint of self-assembly because they're just kind of organisms which are just complex enough and simple enough that we can really, really understand them. So I was rehashing my slides, okay, because I was like, I want to give this talk again, and I didn't want to do something. I decided as a general principle, it's better to speak about what you know than what you don't know. What I don't know, I'll get to at the end, but what I do know is these models that I built. And the last time I gave this talk, I think, was in 2017, and there were 4,000 over here. Okay, so now this number has been updated 5 times since I made my slides. And so this was at 7 a.m. this morning. If you check right now the NCBI database, you're probably going to get so it's like every day people are just uploading virus sequences. It's probably like at this point, it's probably gone up, ticked up by a few. Okay, so there are estimates that there are 10 to the 31 kind of viruses, most of them in the oceans, and 10 to the 7 species. So we know that there are actually, most of the viruses actually prey on bacteria, but we know that, just vertebrate viruses, there are, already like 1 million of them. So one of the things that we know about these viruses is that many of them have very, very short genomes. Okay, so a typical sort of simple virus with single-stranded RNA typically has genetic sequences of only like 1,000 to 10,000 base pairs. And so in the mid-1950s, so this is kind of what Casper and Klug realized, structural symmetry, the shape of the virus, is very well matched with genetic economy. So somehow it just fits just right. And I want to give you a feeling for that. So viruses are not living. They are kind of coexisting with bacteriophages or living organisms. And the simplest viruses, so I'm really using simplicity as a metric here as a mathematician, consists of a genome, so RNA or DNA, and it's contained within a protein shell. Okay, so that's the virus. What the virus has to do is to just reproduce itself. And so what the virus does is the capsid, the shell containing the virus, disassembles when the virus infects a host. Then the genome hijacks the host machinery to make new genome and protein, and the capsid reassembles and packages the genome. Okay, so it's a very, very kind of simple to state reproduction cycle. So I just want to tell you the story of MS2 because this is the virus that we really understand. And so it's an icosahedral virus with a single-stranded, with a single-stranded RNA. So E. coli lives in your gut. So this is a virus that lives on kind of works with the E. coli in your gut. It's the first genome to be isolated in 1961, and it's the first genome to be completely sequenced in this period. So this is really like, in molecular biology terms, really like the first stuff. The genome has these many nucleotides. So there are 3,569 nucleotides. And these nucleotides code for exactly 4 proteins. So 2 of these actually relate to the coat, the capsid. The lysis enzyme is the enzyme that breaks down the bacteriophage's cell walls. And the replicase is the stuff that accelerates the reproduction of the genetic material. Okay, that's the genome. Okay, so it's known. And we know exactly what these pieces in the genome do. So this blew my mind as a mathematician, right? So of course, now I look at NCBI and I just see oh my God, there's all this stuff out there. How do we ever understand that as mathematicians? But I'm like, okay, let me try and understand this little bit as a mathematician. Okay, so what we know about the capsid is, so you've got, if I think about this as a computation, as a kind of assembly language program, so somehow there's just this code, it's supposed to build a shape. What it does is you form many, many copies of this protein. You form kind of one copy of that protein, and these 2 are just to accelerate the reaction. So the coat protein, there are 180 copies of the coat protein. So that's a kind of magic number. There was a kind of quantization found by Casper and Klug, which I'm not going to get into, but it's super simple. And what happens is that the coat protein actually exists in 3 stages, in 3 conformations. And 2 of these conformations form a dimer, so that's those 2. And the other one, so there are 2 dimers. There's the AB dimer and there's a CC dimer. So that's, that's one artistic rendering of what the MS2 looks like. And this one is another artistic rendering, which kind of reveals its mathematical structure. And we actually know that the shift, like the change in conformation between these proteins, is actually triggered by a very specific kind of nucleation event. So let me go back to Casper and Klug, because it's kind of their framework that was used to understand these processes. And they were thinking very much in statistical mechanics terms, like you've got some energy landscape, you are going to go to the minimum of the energy landscape. Well, it doesn't quite work, right? So somehow if you do basic equilibrium stat mech and you try to make an estimate of the time to reach equilibrium, it does not work. Too much time. So this is a kind of well-known problem that goes a long way back. But more specifically, somehow the idea that the capsid would just be the shell that would form itself was disproved by Klug, and that was kind of the work that led to his Nobel Prize. And it's kind of the RNA is playing a role in how this organism is reproducing. And so the sort of work that I'd like to show you is kind of the mathematical structure that underlies this. And this is really lovely work by Raidun Twarok, who's a geometer who kind of reinvented herself as a virologist. And what Raidun's work, with her co-authors shows is that somehow there's this lovely structure underneath where you've got the shell which is being formed outside, which has a polyhedral structure. But in fact, it turns out that the RNA forms with a kind of complementary Hamiltonian path. So I've tried to kind of represent it with the styling diagram. So here, imagine unwrapping the shell that I showed you. And then there's a kind of complementary Hamiltonian path that passes through every single one of these nodes. And it is exactly this Hamiltonian path. When you fold up, that's the structure of the RNA. And this is super important. The use of this structure was super important because when you're actually looking at how, if you're trying to use bioinformatics to actually find out how this thing folds, you need to compute potentials related to secondary foldings. And you don't know it. There's the kind of first folding site that you can pick up. The other stuff is very, very weak, but it becomes stronger as the kind of as the virus and the shell forms itself. So when we look at something like this, we recognize that the kind of simplistic models, the physics-only models, the stripped-down interactions, they don't capture this, right? Like biology is far more sophisticated than the kind of stuff that we're designing in the lab. But nevertheless, we see, as a mathematician, I'm filled with tremendous optimism because I see somehow that nature has solved a kind of very sophisticated mathematics problem, the formation of a Hamiltonian path through, somehow evolutionary means. So I want to take the story and kind of go back. Great. I think I'm on a low battery situation here. Okay, let's do it the old-fashioned way. Okay, so I want to go back to what we did and just tell you a little bit about the polyhedra, like what was the framework that we used. And when you're designing experiments, of course, the shapes look the same. But what you're really looking for is to go beyond that to kind of get a quantitative understanding of two different models, to find unity at the level of models. So David's experimental technology is like, okay, I take a polyhedron, I cut it up, and I let it go. Okay, but some of these fold and some of them don't. How do you know what works? Okay, so you start with a simple shape, the cube. You cut it up. Well, these are the number of ways you can cut up a cube. You give it to an undergrad and say, like, go into the lab, test this. Okay, so a student takes all of these shapes, patterns them all in a way that, it's all kind of coming on the same sheet. She runs the experiments, and you find that certain of these nets, so this particular net does a lot better than that net. Okay, so the beautiful thing about these experiments is you can see the process of self-assembly. So the difficulty when you're kind of like looking at self-assembly of stuff like viruses, you can't see what's happening. You can't see how fullerene kind of forms itself. You can't see how stuff at the mesoscale forms itself. And so these are kind of optical microscope images that show you how the cube forms itself. And in particular, you can kind of match these images that you see with the kind of quantitative, discrete, or whatever, the discrete geometric algorithm that you begin with. And you kind of begin to pick up patterns on how this stuff works.
Quick
so,
question.
Yeah.
If we're allowed to ask questions.
Yeah, please do.
In the previous slide,
Yeah.
I just want to make sure I understand this because this is
Yeah.
really cool. So with your figure, the x-axis are the different types of nets.
Right, and so what's happening over here is it's the yield. Yeah, so these are the different types of nets. Yeah, let me go back.
Yeah.
Yeah.
Right, okay. And then the A, B, C, D are the colors in that figure? What are those?
Oh, so that corresponded to kind of like what you do is some of them form, some of them don't form. So you grade the ones that form, and you're looking at, so in order to get the yield over here, so it's kind of like E means the really good stuff, D means the junk. So like most of these folded well, some of them didn't. And when you go down over here, very few of net 8 folded well, and most of it kind of failed. So it was like you do an optical sorting after you're done, because you often get plenty of misshaped stuff. And so you're kind of looking at which ones form right.
And then the folds that fail, they don't achieve this closure.
Yeah. So like so somehow this thing has to synchronize right? It's like, imagine, so what's going on is it's as it comes close together, it sticks. And so if it comes to a stoon, it kind of gets twisted. So there's stuff that doesn't quite work. And in some sense, like, let's go back, because it's relatively easy to get intuition for it. So this is the one that works really well. And things like this is like things like this don't work, or this one really works, doesn't work very well. So you can kind of see that the stuff that's long and floppy isn't quite coordinated right. I played around with toys to really understand this. But what happens over here is that these two pieces fold first, and you get something which is really rigid, and it just locks into place. So that's kind of how it works. Okay. So I have to thank the NSF here, because what actually happened was that, so David wanted to build the next complex shape. Okay, so the Platonic solids have been classified for 2,000 years. So if you want to test which is best for the cube, well, there's 11 of them, and there's no problem, right? Like, give it to an undergrad, like, go try it out in the lab. If you want to build something a little bit more complicated, like a dodecahedron or icosahedron, like, good luck getting like research funding to kind of run a massively parallel experiment, to test every single one of these nets. So that's a huge part of anything that's happening in this area. You've got this combinatorial explosion of the number of ways in which you can build something. So you need some sort of principled way to actually begin, to choose the right thing. So what we really did, so in terms of kind of explaining the first work that we did, building the more complex shape over here, what it really entailed is actually breaking apart these more complicated polyhedron. They're not all that complicated. And then trying to kind of filter through, kind of search through the space. So it's an optimization problem. And it was a very, very tractable optimization problem. It was a kind of fun problem. I actually did it with undergrads. And that's how we actually designed the shape, the net that was used in the labs at Hopkins. So at this point, I began to get really, really intrigued. And I was like, okay, we were able to build stuff, but why did it actually work? And at the same time, I wanted kind of harmony, let's say, with the models that had preceded us. And so I was like, okay, I want to think about, I know that there are discrete algorithms for thinking about the virus assembly. So let me make a discrete algorithm for thinking about folding. And so here's the algorithm, right? Like, imagine cutting edges and unfolding, flattening as you go. And it turns out, right, like, combinatorics kind of works out nicely. It turns out that this kind of discrete evolution algorithm, so a discrete way of thinking about folding, has several clean mathematical interpretations. So, Bernard's talk on the first day had this kind of lovely kind of networks, that lead biochemical networks that lead to the tomato. And Adria had this kind of even more amazing kind of metabolic network. So here's a metabolic network that I computed. Like, this was like a mistake. I should have written a computer code. I didn't. So I began to do it by hand. And 3 weeks later, I was still doing it by hand. And so what I'm showing you over here is a kind of pathway for folding a net. So in the image, what's going on when I kind of draw an image this way, what it means is that, so if you just take that edge over there, the DEA, what I've done is I've kind of folded all 3 edges. So my algorithm is kind of, I really should have bought my toys. So I've folded up. I've made a corner over here. So I'm making these corners, and I'm kind of moving along. Okay, so that's just the visual representation of what the intermediates do. And then there's stuff like that. Okay, so pathways are always, always complicated, even when you're looking at very, very simple geometric rules. But nevertheless, we persisted. And it turns out that when you're building a cube, this is kind of what you get. Okay, so at this stage, I was like, this is it. Like, this is game over. There is no math over here. And so I was like, okay, I need some way to filter out this information. And a very crude metric was to actually look at the distance moved as you go from one intermediate to the other. You're actually sitting in 3-dimensional space, and you just compute the distance. And suddenly, this thing kind of declutters. So there's a kind of concentration. Like, this is a kind of mathematical word that we would use. And what you see is, out of all of this clutter, a kind of few pieces pop out. So there's a particular intermediate, this kind of rigid intermediate that corresponded to the kind of simple net for the first experiments. And the pathways are kind of collapsing in an interesting way. And then we could test this. And okay, test in a visual sense. You've kind of, you look at the set of all folding pathways, you kind of focus on optimal pathways, and then you test to see if you actually see these pathways in the lab. So it was a very nice collection of experiments, and it left me kind of wanting more. Somehow, when you have a mathematical collaboration, there was a part of it which was really, really fun, just kind of working with the labs, doing kind of, being a kind of mathematical service provider, shall I say. And then got to a point where I actually had real math, by which I mean that there was kind of real territory, like a real mathematical problem that I was like, this is not a solved problem, and I really need to focus on the problem. So this problem actually relates to the computation of rates. So there's a, a clean part where you just kind of break it down into each of these pieces. But any one of these intermediates, right? So if I take something with a certain number of phase glued, it's actually sitting in three-dimensional space. It's a linkage, and it can jiggle around in a zillion different ways. So there's a precise mathematical term for this. It's called an embedding problem. And so somehow, at this point, I really want to kind of nail this down and understand how these pieces are fluctuating in space. So I guess this is for the mathematicians in the audience. So it turns out that at this point, there's actually real mathematics in the sense of some stuff that we don't understand. So we don't really understand in very precise in sort of minimal terms, just algebraic geometric terms, how to kind of describe fluctuations. And there's also simple stuff, and you really need to do that because it turns out that there are very, very simple ways of folding, which are very simple ways of actually forming networks together, which actually have singularities. So if you take something, these are really children's toys, right? But if you take this little arrangement of 6 squares, there's one way of folding it where it looks like it's got 2 degrees of freedom. Sorry, it should be 1 or 2 degrees of freedom, not 2 or 3 degrees of freedom. Sorry about the typo. But there's one where it just becomes rigid, so it can fold this way, and there's a kind of ripple. There's a way that it can fold into. So as a mathematician, you could cheat and make this add some elasticity and stuff like that. But I don't want that. I want a kind of absolutely clean kind of mathematical resolution of how I pass through the singularity when I'm fluctuating. Okay. So this has, at this point, it's kind of become divorced from what's happening in the labs. But I just want to point out that one of the things that you're looking for as a mathematician when you make a collaboration is you're looking for new problems, like new stuff that's intrinsic to mathematics. And this one was one of these problems that popped out. Another kind of challenge was, look, it's a theory, right? And we had a kind of early success. We were able to build stuff. You were able to publish it in a fancy place, et cetera, et cetera. But one of my concerns over here was, is this actually a testable theory, right? So the theory is very straightforward. You want to build an object by attachment of faces. So it's a simple symmetric object. You can break it down into a graph, easy problem. But you need to add rates to this graph. So you've got this gigantic graph, and you have to associate weights to every single one of these. So in principle, if you want to have an experimentally testable theory, you need to have an experiment to test for each of these rates. So for example, as soon as you try to build, so these are very, very they're not particularly complicated shapes. But when you look at the number of edges, you have to think about these as a parameter that must be determined from experimental measurement. At that point, you're like, well, it's neat mathematics, but it's not so clear that this is really a testable way of kind of describing an experimental theory. So I kind of stopped working this field after this for for several years, and I kind of went on to do some other math. And in some sense, now the stuff has been sitting with me for a little while, and so this is the speculative part of the talk, the does life compute part. And I kind of want to put together a bunch of different ways in which we think about the process of building shapes and the way we think about, or the way we should think about self-assembly. So one of the things is that there's progress in math, and polyhedra, like Plato, was awesome, but that's 2,300 years ago. And there's new stuff that we know. So the question that I'm really asking, in some sense, is for harmony. So I want to do good mathematics, I want to do good biology, and so somehow I want fidelity to both disciplines. And so one of the things that the working with symmetric polyhedra revealed to us is that there is absolutely well-defined mathematics to be discovered in this area. So there are well-defined assembly diagrams. The Brownian motion of linkages is an interesting problem. The understanding of sequence-specific folding pathways, the work of Twarok, that's a lovely problem. There are beautiful tiling problems in there. So another thing that Rydon did, which was kind of amazing, is to see that there are kind of forbidden viruses in the Caspar-Klug theory, and these correspond to Penrose tiles. So there's stuff that mathematicians found, which somehow viruses have known. So the tension is really over here. So there's the exquisite design of biology, and then there's the idea of what can humans do. Okay? There's a theory for simple viruses, but viruses are a very, very small part of the problem. Okay? And so, on the other hand, the construction of shape is something that mathematicians have thought about for 2,000 years. So somehow, what you're looking for is some sort of meeting point between all of these viewpoints. So if here's a polyhedra that maybe you guys didn't know about. So this is a little polyhedron invented by Isidor Hafner, who I think is a Slovenian mathematician, just for fun. And so, if you can build polyhedra, somehow one of the things that you should be able to do with polyhedra is to build machines. We were never really able to execute this. And it really relates to kind of a technology where you are sticking stuff together and forming rigid edges than having something which you can kind of jiggle-jaggle. I think this should be feasible at some point, but it was definitely not feasible a few years ago in David's lab. The other thing is that biological machines are not polyhedra, right? So, what I'm kind of showing you over here is a kind of spectacular and absolutely fundamental biological machine, biomolecular motor. It's called the Circa pump. So it's a pump that lives in it's a pump, so it kind of lives in a membrane, and it's called a P-type ATPase. It lives in the membrane and maintains a membrane potential. And in a sense, the reason for focusing on this particular pump is that it's a kind of fundamental molecule, and it's one which is just slightly more complicated. It's just above the FOF1 ATP synthase, which is one that we kind of reasonably understand how it works. It's a kind of beautiful rotary machine. So what actually happens when you've got proteins is that they actually do stuff. It's not that they live in one conformation. They're actually moving around between different conformations. Very distinguished scientists kind of dedicate their entire lives to the understanding of one of these specific proteins, like doing very detailed crystallographic analyses of how these pieces fit together. The good news is that, at least as far as, one of the things that we know, a little bit like MS2, is that you can do sequence analyses, and sequence analyses are very closely tied to preserving the essential geometry of the protein. So there are many different types of ATPases, or many different types of these pumps. There are substitutions that occur across different domains of life, but nevertheless, the fundamental shape, the way this thing moves around, that seems to always be preserved. Okay, so I mean, this is just, again, a kind of proof of concept type picture, and it's kind of, here are ATPases that are kind of conserved across different domains of life, and their shapes are conserved. So in a sense, there are just these kind of building blocks, right? And evolution just keeps using and reusing these building blocks. And so, of course, there's sort of a magic in terms of how these pieces fit together. So I began to think very, very carefully about computation. So, I shifted out and I kind of began to think more and more about AI. And so one of my favorite papers is by Bennett. It's a paper on the thermodynamics of computation. And I was initially trained as an engineer. When I was an undergrad, I had to work on steam engines. So I actually had to do steam pumps and things like that. And so I've always loved thermodynamics. And so I love Bennett's kind of statement of what is a computer. So Bennett's definition of a computer is a computer may be thought of as an engineer that transforms free energy into waste heat and mathematical work. Okay. So what exactly is mathematical work? Well, one way of thinking about mathematical work is as the evaluation of a Boolean function. Okay, so when you're building a computer, like if you're going to do evaluation of a Boolean function, then you have to find a physical system that will evaluate Boolean functions. This is a problem about logic gates, and so you can cook up different ways of making logic gates. And so in Bennett's paper, he has a bunch of kind of loopy designs of computers. So one of them is, what could be used as a model for optical computers. This is by Fredkin and Toffoli. The other one is, it's kind of related to what I just showed you. It's a hypothesized, it's not a real enzymatic machine, but it's something that, Bennett is cooking up. And here's something else that he does. So he was interested in the idea of reversible computation. So really computation in the presence of noise. And he came up with something that he called the Brownian Turing machine. Okay, so none of these are actual computers, like none of them have actually been implemented. But as proof of concept, like what is computing? What is mathematical work? It's a kind of lovely framework that tells you, like, okay, here are fundamental limits. Here is how you should think about computation. There's another purely mathematical. So this is a purely mathematical construct of computing. And this is the work of the philosopher Hao Wang from the 1950s. And this is a beautiful paper, popular paper from the Scientific American in 1965. And what Wang does is he shows that logic gates can be realized as tiling problems. Okay, so the game here is the following. You invent a bunch of tiles, okay? And the rule is that you can put two tiles together when they've got complementary edges. So, I can join the A and B, I can join the B and the C, or I can join the C and the A. And when you're doing this joining, what you're really doing is you're making a logic gate. So that's the first step. And then Wang did something utterly brilliant. And he basically said that a Turing machine, or most specifically decision problems, can always be made equivalent to a tiling problem. So they're like, okay, there's this kind of abstract world of Turing machines, but here's a concrete world of a Turing machine. I will make a set of tiles. I will give you, you want your computer program, I will associate to the computer program a set of tiles. And if you can tile the plane with those tiles, then your Turing, you don't have a halting problem. And so somehow there's a, let's say, theoretical computer science framework that says that decision problems can be equivalent to tiling problems. So this is very, very nice, because what's actually happening is it's giving you intuition, geometric intuition, for how computation actually works. It was kind of, in the late 1990s, it was realized in absolutely spectacular experiments using sticky DNA by Ned Seaman, Paul Rothman, and Eric Winfrey at Caltech, kind of leading to the field of DNA origami, which I'm not sure if this is what Rebecca is talking about, but it's just this absolutely thriving field. But it kind of originates in a kind of pure mathematical construct of what is a Turing machine? How do I see it visually? So on the other hand, there's a different type of human mathematical work, and a different type of human mathematical work, going back, again, 2,000 years to the Greeks, is how do you build a geometry? So as mathematicians, you set very rigid rules on yourselves. I will do ruler and compass constructions. So that's for, the world of logic is very different from the world of geometry. And so here's beautiful images from a paper of kind of Thurston Weeks from Scientific American. And these are geometers. And so what geometers are really thinking about is how do you build shapes? So how do you build manifolds? That's the real problem. And so what you do is you can think about manifolds as being defined by building blocks. So that's the kind of basic patch that you're looking for. But this basic patch you can think about as coming from a linkage. It's the configuration space of where the linkage goes. And then you glue it together in a particular way. So I like this a lot because, of course, this is kind of the picture that's living in my background when I'm thinking about even the self-assembly of little viruses. I'm thinking about how these shapes are sitting in space and how they're jiggling around. So I've got these linkages are moving around, defining configuration spaces, and that's kind of how I'm building my complex shapes. So what's happening at the end is I'm actually trying to reconcile two different ways of doing mathematics. So there's a kind of world which is based on logic and computing, and there's a world which is based on, let's say, calculus and intuition, ruler and compass constructions, which is geometry. And so one of the kind of, so when I say does life compute? So what I'm really focusing on this one specific problem. Like I give you sequence, you tell me shape. Well, clearly this is what AlphaFold does, right? It's a computation in every sense of the word. And it provides an astonishing kind of resolution of a fundamental bottleneck in molecular biology. So the question I'm really asking is kind of what would it mean to understand how it works, right? Like we've got this amazing enabling tool. How do we understand it from within? And so what I'm giving you is kind of a way of how mathematicians think about it. And I think about mathematics as a kind of human form of collective intelligence, kind of accumulated knowledge that's kind of passed on and kind of survives over these very, very long periods. And so there's human-tested knowledge, right? So kind of when we're using Platonic solids, it's not that I really care about the rest of Plato's life. Well, actually I do as a human being. But somehow all that matters is this kind of abstraction that transcends time. So one way of thinking about mathematics is as the study of reproducible mental objects. We all there's a Platonic reality, and we kind of realize this Platonic reality. And one way I'd like to think about molecules like the ATPases is that these are kind of reproducible biological objects that can be reused by evolution in many, many, many different ways. So somehow, when you make a mechanistic understanding of self-assembly, just beginning with these simple shapes, you actually discover very interesting mathematics as you go along. And you realize that you really need fairly sophisticated mathematics to actually get to an understanding of more complicated stuff. So I put the stuff away for a few years, and somehow now I was in kind of mathematical monastic territory for a few years, and now I'm kind of coming out of that. And I'm like, I want to go back to this problem, and I want to go back to this problem because I think these frameworks which are tested are, in some sense, one of our best ways of trying to understand, without doing too much computation, what's actually going on the hood of one of the most important developments in science with AI. But I think the AI over there is also tied to the collective intelligence of kind of the human mind, and somehow that's what I really meant when I said, does life compute? So thanks very much, and so let me stop over here. I hope I'm not over time. Yeah, awesome, thank you.